Welcome, dear students. In today's session, we'll talk about prokaryotic cells. Prokaryotic cells basically include two large groups, that is archaea and bacteria. Our primary discussion today will be focusing largely on bacteria. Okay, so bacteria come in various shapes as well as sizes. We are primarily focusing in this particular image on the shapes. Bacteria can be spherical, they can be rod shaped, they can be spiral shaped, or they can be comma shaped or even other shapes as well. So again, within spherical structures or even rod shaped structures, cylindrical structures, what we often find is the arrangements are also different. A single sphere can be present on its own, which is often uh, usually called as monococa or monococcus. Then you can also have cells arranged in the form of chains, with which we use the word streptococcus. You can also have these structures arranged in the form of a bunch of grapes, etc., with which we use the word staphylococcus. Okay. So similarly, in the case of your bacteria also, you can have a single individual rod-shaped bacteria, and often you can also have chains of bacteria as well as present. Then the spiral bacteria we saw in the starting as well, that is Treponema pallidum, the causative agent of a sexually transmitting disease called as syphilis. There is another shape that is not mentioned here, that is a comma shape, very classic to a microorganism called as Vibrio, in particular Vibrio cholera, which is the causative agent of an extremely deleterious disease of the intestines, that is cholera, often spread through contaminated food and water. Now, let's look at the basic structure of a prokaryotic cell. Prokaryotic cell is composed of a cell wall and a cell membrane. The cell wall is different in certain bacteria, which we'll talk about in upcoming slides. Inside the cell, we do not see any particular organelles or membrane-bound structures. We do see certain invaginations of these membrane, which are called as mesosomes, which houses several enzymes and increases the surface area internally, which usually are also predecessor to the kind of imagination we will see in the case of plastids and mitochondria as well in the case of eukaryotic organisms. But in general, we do see these folds, but not any particular organelles as such inside of microorganisms, such as bacteria. We do see a centralized DNA or genetic material, which is called as the nucleoid, which is surrounded by ribosomes. So in this case, the entire process of DNA to RNA to proteins is entirely completed within the cytoplasm only. There is no demarcation of these functions happening as such. And we also often see these flagella, which helps the organism to move. And based on the structure of the or the location of the flagella also, we can categorize bacteria into various groups as well. Whether the flagella are located on one end, whether they are located on the side, whether they are present on both ends, and so on and so forth. Do also search about any other features such as pilla and fimbria and what do they help a bacteria in. These are also certain structures which are present around certain bacteria, but they are different from flagella. In what ways? Do find out, and also do find out how pilla and fimbria themselves are different from each other. This is another image of the bacteria showing in much more depth in the certain parts. The outer structure that is shown here is a capsule, which is often found in many microorganisms or many bacteria, especially infection-causing organisms such as the Streptococcus pneumoniae. These capsule helps the organism to prevent damage from the immune system. So it's a counteractive measure that has evolved in bacteria to survive easily inside of other organisms. Then below this capsule, there is the cell wall and the cell membrane. You can see the flagella starts originating from the cell membrane and not the cell wall itself. It might be embedded partly in there, but it starts from the cell membrane. You will understand which structures are making up a flagella in more depth in the subsequent sessions. In certain bacteria, the cell wall could be very thin and it might be surrounded by another layer called as the outer membrane. These kind of bacteria, which usually have a thinner cell wall, are called as gram-negative bacteria, while those which have a very thick cell wall are called as gram-positive bacteria. There are bacteria such as tuberculosis causing bacteria, the Mycobacterium tuberculosis, which also have additional layers of glycolic acid and other materials, which does not allow the gram stain to grow very easily inside of the cell wall, due to which they might confuse us to be gram negative, but they are really not. And there are also organisms such as mycoplasma, which may lack cell wall altogether. So we cannot say them either as gram positive or gram negative. Research about mycoplasma is quite interesting, not just based on the cell wall composition, but also their size. So this is the image comparing gram positive and gram negative bacteria. What is common to both of them is a plasma membrane, whose length is almost equivalent in both of them. But what is unique in them is the peptidoglycan length. Peptidoglycan, along with certain other carbohydrate protein bound molecules, compose your cell wall. Peptidoglycan is the most important component and usually made up of carbohydrate unit with a peptide attaching two different chains of carbohydrates. This particular structure, the peptidoglycan, is thicker in the case of gram positive, due to which the gram stain that we use, crystal violet, often embeds itself inside of these peptidoglycan cell wall, but it is easily washed out in the case of gram negative bacteria because there is no space for it to retain itself. There is an additional layer also present in the case of gram-negative bacteria, which is called as the outer membrane, also at times referred to as the LPS, because of the composition, that is the lipopolysaccharide layer. So basically, we in the gram staining method, we use two stains, the primary stain and the counter stain. The primary stain bound within this peptidoglycan cell wall, making the gram-positive bacteria appear purple to violet, while the counter stain, usually safranin, binds with this, crosses through this thinner peptidoglycan cell wall and making them appear red through this stain safranin. So that is the gram staining method. Now let's talk about what capsule and slime layer and biofilms are. Both capsule and slime layer are extra layers which are found usually outside your cell wall. And these is the best known example is that of Streptococcus pneumoniae, the causative agent of pneumonia, the disease in which the fluid fills inside of your lungs due to the infection. And this encapsulated gram-positive bacteria, Streptococcus pneumoniae, use this capsule for protecting itself against any harmful effects of the immune system. If this particular layer is quite disorganized, it is called as a slime layer. If it is well organized and quite stiff and tough in nature, that is called as a capsule. Now, biofilm, on the other hand, is a deposition that happens around certain bacteria. The best example to understand biofilm is what happens on various surfaces, and especially your teeth. Whenever you do not brush very frequently, you might have experienced when you eat too much of sugary food, you might have experienced that a yellow color deposition happens, which is initially called as a plaque, which later on stiffens up to form tartar. So this dental plaque is nothing else but bacteria, also surrounded by various fibers that it has accumulated from the surroundings, and that is through the components of your saliva or from the food materials and also certain waste material that it has released outside. This entire mixture creates a protective environment for the bacteria to increase in number continuously without any damaging effect of your secretions that are released outside. So basically, it forms a fortress kind of thing where the bacteria can live easily, multiply, consume food, and get easy access to your teeth, etc. And at the same time, no other chemical that is being produced outside can enter inside, thus protecting the bacteria very easily. So that is what biofilms are. It's a more of community-driven effort by various bacteria working together to utilize the resources well and protect themselves from immune system. This is the image of a bacteriophage, which is a type of virus that infects bacteria. So that is the most classical shape that you often see of a bacteriophage with a head kind of structure, this body, and with a claw kind of structure, which are visible quite here. Then they inject their DNA inside of it. The DNA gets incorporated, and then uh, it becomes part of the DNA of the bacteria and multiplies with it. Bacteria also have two types of DNA. First is your main central DNA, and then is your plasmids. These plasmids provide space for certain type of genes which are often responsible for, say, antibiotic resistance or any protective situation for a bacteria. So this particular image shows that of a T4 bacteriophage injecting its DNA, and then various sections and close-ups are that is shown of the bacteriophage and how it is injecting the DNA content, which can be seen getting transmitted from here to here. The structure turns from dark gray to a very pale white, transparent kind of nature because the DNA has been inserted inside now. Okay, so it has emptied all of its DNA into the bacterium. So through that, we have understood in depth about what prokaryotes are, what are the basic important structures within them. But that's not enough. Prokaryotes is a very beautiful word, and they are way beyond diseases as well. Many of these bacteria play a very important role in helping uh, to shape our environment, form several interactions with various kinds of environment, and such as processes such as decomposition, recycling of nutrients, etc. So they have important roles beyond just being problems. So do search about them, even about viruses as well, because viruses also play a very important role beyond just being disease-causing agents. They do help in population control, and they do have a role as such to maintain the ecosystem as well. So do try to expand your horizon of understanding organism way beyond this human-centered perspective and dwelling more into depth of how organism and broadly our nature works. Thank you, and I will meet you in the next session about eukaryotic cells.